Okay, good morning everybody. This is day 3 of the Public Health Applications course. Um, we've got a lot to go through. Today is a busy day because we're basically going to try to give you the entire picture of how to assemble distributional pictures for individual species. Now, to begin with, we've done a niche modeling course. And so definitely check that out if you're going to get into the specifics of this. But what I'm going to try to do to start us off is to give you an overview of the niche modeling process and some commentaries about specific points where we may be amending methodologies essentially now. In view of recent research. And so that's what I'll try to do in this first hour. Then we go to Yoshi Nakasawa in Mexico City, who will give us a worked example. Then we'll come back and I'll give you a quick overview of kind of the particulars of disease systems. Then we'll go to Luis for more particulars of disease systems. And then Luis will talk about evaluating risk maps via independent data. And then we'll have some time for questions and answers. As always, hit us with questions. So I've got a lot to do in this first hour, so I'm going to jump right into it. And essentially, here's what I want to do in this first hour. I want to give you a broad overview of the niche modeling process, and I'm splitting this into five pieces. So that's kind of the outline of this first hour. First comment is there's a whole course on niche modeling or distribution modeling, if you prefer. That link takes you to a week of videos on this topic. Of course, that 3 of us gave back in February of 2013, so I recommend that to you highly. So my basic comment is that niche modeling has perhaps gotten a little too easy. There's very easy access to lots of occurrence data, easy access to geospatial data, easy access to modeling tools, and there's lots of literature out there that sets out examples. Sometimes maybe we ought to be thinking that it's too easy, which is to say things can get so easy that we stop thinking about them. And so that's essentially the commentary that is underlying this whole talk. So let's start with that first step of accumulating input data. And what I'm going to do is I'm going to show you these orange slides that show you kind of a flow diagram. And that's going to be in these 5 pieces. And then for certain parts of it, I'm going to give you more detail. So for accumulating input data, we start by collating primary biodiversity data that document occurrences of our species. And that can come from many different sources. It might come from GBIF or VertNet for biodiversity. For disease occurrences, it might come from a public health institute in your country, what have you. And definitely a first step is to assess spatial precision of those data and throw out the data that are least precise spatially. A third step is that of setting aside some data for model evaluation. And we'll come back and talk about that later. On a different front, you need to collate GIS data, essentially raster data layers that summarize relevant aspects of the environment. You're going to assess the spatial autocorrelation in those data to make sure that they are relevant and informative at the spatial resolution and extent that you're working at. And then you're going to hopefully process those environmental layers to be kind of maximally relevant to the species that you're working with. we're going to talk about several of these steps in more detail. So the occurrence data, the goal is to represent the full diversity of situations under which a species maintains populations. Spatial biases are okay. We know that biodiversity data clusters along points of access. Usually it's roads. But what is really, really bad are biases within environmental space. If you are not sampling, or whoever accumulated the data is not sampling the wettest areas or the driest areas or the hottest areas, then that will translate directly into biases in your niche model. So more data is usually better, but not always. And we need better data instead of more data. So this is an example from Brazil with the speciesLink network. And this is just to show you the huge accumulation of occurrence data. So along the left y-axis, you can see the number of records in the speciesLink network. The green area is the accumulation of, the green and blue areas are the accumulation of data online overall. But the blue area is the portion of that that is georeferenced. So what you can see is that during the entire history of the speciesLink network, it's been just about half of the data, maybe a little bit less than half of the data, are georeferenced. So georeferencing is an important process where we add geographic coordinates to a biodiversity record. And a very important step is speaking to the uncertainty in those geographic references. And here I've just inserted some very simple illustrations of what uncertainty means. If I say northeast of Lawrence, Kansas, I'm really not saying exactly 45 degrees to the right of north, but rather I'm saying kind of somewhere between north-northeast and east-northeast. And so that immediately gives us some uncertainty about the direction. And if I say 8 kilometers, I'm not talking about exactly 8 million millimeters. Instead, I'm talking about somewhere between 7 and 9 kilometers, or 7.5 and 9 kilometers, 8.5 kilometers. So we have to essentially integrate over all of these sources of uncertainty to generate a measure of how precise our data are. And so a georeferencing step, which you may have to do if you need that half of the data that is not georeferenced, and half may be a high number when you look at the broader world of biodiversity information. But georeferencing needs to represent the place where the species was found as best as you can. But it also has to represent the uncertainty with which that place is characterized. And then, kind of in a bookkeeping sense, it should summarize the methods that were used to establish those coordinates, establish that place, and it should probably preserve all of the original information in case you need to reinterpret the georeference at some point in the future. And so when we do this georeferencing step, we need to do it with an eye to error. So here's a data set for a genus of birds in Chile, and there's some things that I want you to just look at. I want you to see, for example, this point, which is this southern species. See, there are 3 species, 1, 2, 3. But notice that the yellow diamond species is also represented by 1 point way up in the distribution of the northernmost of the 3 species. Or amongst the southern species down here in Patagonia, in the right-hand panel of this of this figure, you can see 2 points that seem to be well south of the main distribution of the species. Those are things that are worrisome. Doesn't mean they're wrong, but it does mean that we should look at them. And if we look more carefully, we can see the northern species down here at the border between the southern and central species. We can see the southern species up here in the range of the northern species. Notice that that record is also off the coast. Now, sometimes that's just a result of a coarse coastal outline map, but also sometimes it's just an error. So even once we have our list of latitude and longitude points, we really need to look at our data again, kind of in a quality control step. Here's an example from Kenya with a data set on some plants. And what I've done here is to take the state or district that is listed as the source of each record, and I've taken the latitude and longitude and plotted it on a map of the country. And so look at this northeasternmost district, and right away you see that all of the points in that district have the same district name. That's good and consistent. But look at this northern district, the central northern district. All of the points there are this kind of dull green color, except that there's one purple point in there. So that's a point that had a different district name. So again, those georeferences are not necessarily wrong, but they are records that we should look at carefully and make sure they're not wrong. So that's a data quality step. And we can call that data cleaning, where we're attempting to detect seriously erroneous records so that they can either be excluded or treated with caution when we do our analyses. We can look at issues of internal consistency, like does the latitude longitude match the country or the state or the district? But also we can use steps of external consistency, like do the occurrence data match the known distribution of the species spatially? I could go into a lot more detail on this, and indeed we have a whole course on data cleaning, so I recommend that to you. And I'm going to move on in the interest of giving you a full view of this overall field. Then just one quick comment referring to this paper. This comment is about the idea of quantifying how much error there is in your occurrence data set. And we call that the E parameter, and I'll make this paper available to you so that you can read about the E parameter. We'll be talking about that later. And then remember, we talked about subsetting data, and we're going to come back to this. This is to permit model evaluations. For the environmental data, they need to be in raster format, and hopefully they will be relevant to the distributional potential of our species. More dimensions is generally better, but you have to remember that the more dimensions, the easier it is to overfit a model. So we need to be thinking about the highest number of truly relevant and independent dimensions. The major sources are pretty clear: climate data, remotely sensed data when you have recent occurrence data that are finally resolved, topographic data, soils data, and others. The spatial autocorrelation considerations are important. This is a remotely sensed image on top of a climate data set. And you can see that the climate data set includes thousands of the remotely sensed data pixels. If you're working at continental or global extents, that climate data set might be perfect. But if you're looking at local local extents, then you may need something finer like the remotely sensed data. Spatial autocorrelation causes non-independence in model evaluations, and it also causes inflation of sample sizes. So both of those are pretty important and pretty damning when you try to use a data set and don't take into account spatial autocorrelation. So here's a paper that also we can post for you that talks about spatial filtering as a way of improving model performance. And then last comment is we can take kind of the crude data sets and we can process them to be maximally relevant. And this is kind of a set of GIS exercises, but notice that it gets done all the time. These are just data sets off of the web that were processed to be relevant for example, horticulture or agriculture. What's the average date of the first freeze? What's the earliest date of seeing 28 degrees Fahrenheit, which is pretty cold for those of you in tropical regions? We can develop this sort of synthetic map, which then gives us perhaps information that's more directly relevant to the species of interest. Okay, here's our second panel. You can see I'm moving fast. All of these papers and all of these slides will be made available to you, so don't worry about it. You'll have access to all of this. But essentially, once we've got our input data together, then we have two very critical steps. One is assessing the BAM scenario. Essentially, what is the topology of those three circles that we talked about yesterday in terms of BAM. And we need to come up with a specific a priori estimate of essentially the area that's been accessible to the species and also that's been sampled. So those are M and we could call it S. So this Saup et al. Paper is all about the BAM configuration. And I'm going to run through it very quickly for you. Essentially, you remember the Eltonian noise hypothesis where we allow ourselves to neglect the biotic. And so we end up with this A and M as constraints and not B. And between those A and M circles, we can kind of come up with four different relationships between the two. We can have them overlapping completely or partially, or we can have one nested within the other. Now, what we did in this study is we created some imaginary species, and we created them just basically by hand, and then we created BAM scenarios. So this would be a classic BAM scenario. Our species overall is a desert species in northern Mexico. Again, this is just made up. Its overall A, its habitable area, is what you see in blue and in yellow. But we said, well, what if the M separates the Chihuahuan Desert from the Sonoran Desert? And so we made up a species that could only access the Chihuahuan Desert portion, the yellow portion of its potential distribution. Then, so we did that for a bunch of species, and for each one of those species, we created these four BAM scenarios. And what I want you to see is that for the upper left and lower right BAM scenarios, we were able to calibrate pretty good models. Essentially, perfect models are in the upper left-hand corner of these graphs, and the lower left part of each of these panels, essentially where the axes intersect, is a perfectly random model. And all I want you to notice is that under two of these BAM configurations, you can fit good models. But under the other two, the lower left and the upper right ones, most of our virtual species, our imaginary species, give us models that are perfectly random. So essentially what I'm saying is back here in our diagrams, the classic BAM and Hutchinson's dream scenarios actually can give us usable models. but the other two are very, very challenging for niche modeling. So if your species is in a situation in which you are, essentially everything that is accessible is suitable, you're not going to be able to fit a good model. Let's go on to kind of a next step on this. Here's a paper by Barve et al. And this is a very crucial paper because it talks about what should be your area of analysis. And again, we created a species, just kind of an imaginary species to be able to do these tests. And it has the distribution. Its A is this white area in the middle of this diagram. And then we explored different calibration areas that were bigger and bigger around it. And so we call those 0%, 5%, 10%, 20%, 40%, 60%, and the whole area. Now, all I want to show you is that that calibration area affects everything we do in niche modeling. So if we're talking about calibrating models, this graph in the lower left is a comparison of the maps. And up near 1.0 is that the maps are completely identical. And down near 0.5 is that they're very different. And these are different sizes of M compared to a model calibrated across the whole rectangle. And what I want you to see is that they're not very similar. They're down in the range of 0.5 or 0.6. So right away we see the size of the calibration area makes a difference. If we're evaluating models, which Luis will talk about with you later, we can see at 0, the critical value here is 1.0 on the AUC ratio, and we can see a 0% buffer size is centered on 1.0, and so that's not a significantly predictive model. But the exact same model evaluated over a broader buffer, like 20, 40, or 60%, becomes significant because it's above 1.0. So critical effects of the area, the M hypothesis, on your model evaluation results. And indeed, if we're comparing models, I'm not going to go into detail on this, but if we compare the red area and the green area, both of which are manifestations of the same niche, just over different geographic areas, here the critical area, the critical value is shown with an arrow. And what I want you to see is that at 0% buffer, we can't establish these species as being significantly similar. They're essentially random with respect to one another. But up here at the broader buffers, like 40% and 60%, indeed the entire null distribution is to the left of the observed, the arrow. And so we're able to establish that these two species are significantly similar. Again, I'm not going into the details. I just want you to see that this is affected by the calibration area, the M hypothesis. So this is one of the most crucial and the most ignored steps in this whole process. And essentially what you need to do is come up a priori with a hypothesis of the accessible area for your species. Read that paper, please. We also need to take into account sampling. This is just to give you an example. This is a species of New World oriole that's in the southwestern US and in Mexico. And you can see lots more records up in the northwestern part of its range as compared to the southern part of its range. And that's all a consequence of there being more bird watchers that are reporting their data in the US as opposed to in Mexico. So taking into account sampling and sampling intensity in these models is very crucial. And then last comment on this panel is the idea of reducing dimensionality. And this is essentially creating simpler environmental spaces, maybe using principal components analysis, but simpler environmental spaces so that your modeling challenge is easier. Okay, you can see I'm moving very fast, but this is panel 3. This is calibrating our models. And here what we're talking about is using different niche modeling algorithms. We have to think a little bit about which algorithm. We have to think about parameterizing that algorithm. And we have to talk about thresholding our models. So here's some work that I've been developing with my colleague Huijie Chao in China and my colleague Jorge Soberon here at the University of Kansas. We're talking about essentially how no silver bullets. And this is the idea that there's no single solution that's always best. So here we go again, imaginary species. We set them up so that they would yield different types of challenges to different modeling algorithms. And so you can see these fundamental niches, which are ellipsoids, across an environmental space. What we're doing is we're comparing with this very famous figure from a paper by Elif in 2006, where we are looking at average performance of niche modeling algorithms. And so this was 2 measures of average performance. They're called core and AUC. Really, AUC is the only ones that's used anymore. But essentially, the ones in the upper left-hand corner are the really good ones, and the ones in the lower left-hand corner of this graph are the really bad ones. And so we developed similar plots for a bunch of different algorithms as a function of different characteristics. And what you can see is that in each of these 8 plots, you can see different types of performance. And you can see the different algorithms, which are in the different plots, giving different sorts of performance. But if we look at these data a different way, like for a given species and a different environmental scenario, which algorithm was best, here we can see the frequency with which an algorithm was best and the 1, 2, 3, 4, 5, 6, 7, 8 algorithms arrayed across the x-axis of this graph. And it doesn't matter which is which, and we're not talking about which of these algorithms is best all the time. We're talking about which of these algorithms is never best. And what I want you to notice is that each of these algorithms is best some of the time. So in a no silver bullet world, we really shouldn't always use one algorithm. So it's very common just to see people using maxm or random forests or boosted regression trees. That's not a good idea. What you should be doing is testing different algorithms, see which is best for your particular species, and using that algorithm for that study. That's what I just told you about. Let's talk a little bit about model thresholding. This is essentially going from a continuous output to either a stepped output or a binary output. And what I want you to think about is the relative merit of a presence datum versus an absence datum. So this is a probability tree with 3 decisions that correspond to circles in the BAM diagram. So was the place accessible? Is it suitable biotically? And is it suitable abiotically? But then we have to ask, was the site ever visited by a researcher? Was the species detected? Sometimes the species is there, but it just doesn't get detected. And then we can add in others like, was the detection reported, and was that record shared? But notice that only if you get a yes answer to every single one of these questions do we get a presence record. So that's that top green bubble in the right-hand bar. where there's a black arrow next to it. That is a presence record. All of the others are absences, even though species might have been there. So we really have to think about how much do we believe a presence record. Presence records will generally be true unless there's an error of identification or an error of georeferencing. But an absence record can very frequently be in a place where the species is actually present. In fact, probably that's why you're doing a niche model, because you want to know other places where that species, or that disease, or that pathogen can occur. And yet that's an absence record. So we need an approach that prioritizes omission error over commission error. And I would suggest using an approach that looks first at omission error and then at commission error. So there's a whole unit in the niche modeling algorithm about thresholding, but essentially the steps are to choose an amount of omission error that you can tolerate, and this is related to that E parameter I mentioned earlier, but you choose that toleration of omission error. You choose the highest suitability value that includes that much of the calibration data, essentially 100 minus E% of your calibration data. And that is a very appropriate way of thresholding your data. Okay, last point in this panel is about parameter choice. Sorry, there may be one more after this. But there's this very interesting paper by Dan Warren and Stephanie Seifert. And essentially what they're talking about is using model selection criteria as a way of parameterizing an algorithm like MaxN. A very interesting paper. I recommend you take a look at it. Okay, then we get to model evaluation, and we're going to have a whole lecture on this shortly, so I'm not going to go into a lot of detail about this. In fact, I'll go through the slides very quickly just so that you see them. But this part of this of the process is crucial. A model that hasn't been tested is not necessarily informative. So, in the crudest sense, think about archery. We have a target, and we have two archers. One of them fires a bunch of arrows at our target and gets the result you see in the upper left. And the other of them fires a bunch of arrows and gets the result that you see in the bottom right. Which archer is better? That's essentially all we're doing. We're asking how frequently is our model prediction, which might be the bullseye in this target, how frequently is our model prediction able to anticipate an independent set of test points? And in the simplest sense, we can do exactly that. Luis is going to talk about threshold-dependent approaches. This is the cumulative binomial distribution. And then in a continuous sense, we can talk about threshold-independent approaches. So this isn't a single bullseye, but rather better in the middle and worse outside the middle and worse farther out. Now the problem is that the most common threshold-independent approach is this AUC measure, and as this paper lays out, there are a lot of major problems with using AUC as a measure of model performance. So with a couple of colleagues, a few years ago, I published this paper that provides an alternative. And again, Luis is going to go through this point in much more detail, so I'm not going to spend time on this. Last point about model evaluation is the difference between significance and performance. We definitely want to know that our model performs significantly better than random expectations. That's crucial, but it may not be enough. Which is to say, imagine that we're predicting coin flips, and I have some algorithm that allows me to predict the outcome of coin flips better than random. And so maybe I'm able to predict that 51% of the time. So it's definitely better than random if I have a big sample size. But that might not be enough for the purposes of my study. So we always want to pair a significance measure with a performance measure. And performance measures might be things like omission rates or correct classification rates. So that's a really crucial part of this topic. Now, the last of the 5 pieces of this process are, okay, we've calibrated our models, we've evaluated them, and we've transferred them onto the other situations that we're interested in. Maybe we want to know the potential distribution of a species on another continent. Maybe we want to know how our species potential distribution will change through time, etc. So this is called model transfer. This is a very frequent reason for wanting to do these studies. And I just want to present to you a little bit of work on model transfer and being very careful about extrapolation. So look at this paper by Owens et al. And essentially what we're doing here, remember this diagram I showed you a couple days ago. We have an environmental dimension across the x-axis, and the portion of that axis that is essentially visible to us because it's within the M area, the accessible area, is this white region in the middle. Lower values to the left and higher values to the right are literally invisible to us. So what we don't know is our species response outside of the M area. To the left side, well, we're going to probably be okay making the assumption that our species has a unimodal response to this environmental characteristic. And so I'm okay with basically assuming that the response to still lower values in this diagram, which is to say values farther left, will be 0 or low. But to the right, I've got a problem, which is to say, notice I've got 4 different ways of extending that curve. Look at the upper one called extrapolation. Right at the edge of my M conditions, my response is still going up, so it could be that it'll keep going up. And all the way over at the right edge of the page, my species is still very happy. Or look at the other extrapolation. There, essentially, my model is guessing that it has a nice bell-shaped curve of suitability, and so it comes back down to 0. Or look at truncation. Maybe, just by coincidence, the environmental limit of my M area is also the limit of my species niche. Well, if you just run a Maxent model on default parameters, it actually does something very interesting. It uses this process called clamping. And that is whatever the marginal value is, the value right at the limit of M, that value is held as the value for all values farther to the right. And so clamping, if this was a temperature axis, clamping is going to say that your species is just as happy when the water is boiling as at the environmental limit of your M area. You may not be happy with that assumption. So what we did in this study was to first established that we had this problem of clamping, and then we applied that to a couple of my previous publications, because it's always nicer to criticize your own work instead of somebody else's work. So this is just an illustration, same sort of visualization, where we have, we have temperature and precipitation along the x-axis. The conditions represented within the M area are shown in the white regions, and the extrapolative regions are shown in gray. What I want you to notice, look at the upper left graph, is that we have Anopheles arabiensis responding to, in that case, temperature, but then our model extrapolates all the way down to ridiculously cold temperatures, like 100 below zero, and all the way up to ridiculously high temperatures, like 100 above zero. And our model is just making silly assumptions about the response of our species. Anopheles arabiensis is dead when the water is boiling, but our model made stupid assumptions. So what we did was we developed this metric called MOP, M-O-P, and what that does is that it asks, compared to the calibration region, which you now know should be the same as M, which areas are simply out of range? Those are the really dangerous conditions, because that's where you're going to get extrapolation. And so those areas, look at this 1% plot in the upper left, those regions are shown as black. And we would say that you simply should not interpret any prediction within that area. But then even when things are not out of range, they can still be relatively different from the calibration area. And those are shown in the red in these different panels. Ignore the bottom panel, perhaps. But where you see red is an area where the conditions are relatively different. Perhaps the 1% and 10% areas are the most, maps are the most important ones, and the 50% and 100% ones are less important. But the point is, we might be careful in any of these areas where our conditions to which we are transferring our model are relatively extrapolative. So I'm not going to go into the comparison between MESS and MOP. MESS is implemented within Maxent, but it's not implemented completely appropriately to speak to the issue of model extrapolation. So I would recommend to you MOP as a better mapping of extrapolative conditions. This diagram is essentially another way of looking at where you expect problems. So we have this whole area that we're interested in. That's eta of G. Remember my first talk in this course. We have eta of M. Those are the conditions represented across our calibration area. And then we've put points for two different kinds of species. If you had this species that has the open circles, you're probably okay, because all of those points are well within the conditions represented across M, and all the way around that cloud of points we have areas of conditions represented within M that are not being used by that species. But then if you look at the other species, the one with the closed dots, that's a species that's peripheral within the environments within M. And those are, that's a species where we're going to get into these extrapolation problems. Okay, so that's a whirlwind tour of ecological niche modeling in 5 panels and a lot of studies designed to give you more background. Again, all of those studies will be available to you, but don't let this process get too easy. It's not just an issue of loading up some data and pressing the button. That's when you get into some problems and make some mistakes. So there is the whole process. Again, all of these papers and this presentation will be available to you. So it's 9:45 right now. I don't think we've gotten any questions from any of you yet. Please do send questions because we're setting aside time to answer them, and I'm sure that I did not answer all of your questions with a perfect presentation. So do send us some questions. You can send those, as I've told you before, to biodivtraining@gmail.com. And that email address is on the Google Plus events page. We will be back at 10 a.m. my time, which is 4 p.m. London time, 6 p.m. Nairobi time. We'll be back in 14 minutes with a very interesting presentation from Yoshi Nakasawa on applying these methodologies to monkeypox. Okay? See you in just a bit, and thanks for tuning in.